chapter 14, we are talking about the structure and function of DNA. So we'll talk about the structure, and then based on the structure, we'll talk about how does DNA function. The basic features of DNA are, one, it's a double helix. So double means two sides, and then helix means that it wraps around. It has two sides for, we'll talk about nitrogen base pairing. That you have a base pairs with another base. That's why you have two sides. That's why it's a double helix. And you also have a backbone that is a repetitive sugar, phosphate, back and forth. Those are the main things about the structure of DNA that are important. Let's think back to the first unit. DNA and RNA are organic molecules. They're part of a biomolecule group. Which one do they belong to? Proteins, carbohydrates, lipids, or nucleic acids? Okay, yeah. So in the name, DNA is called deoxyribonucleic acid. It tells you right there the biomolecule group, the organic molecule group it belongs to. And RNA is ribonucleic acid. These are both very important in how we are structured and organized, how everything in our cell is made, and how it functions. The monomers of the nucleic acids are nucleotides, and we're going to look at the nucleotides. You're probably most familiar with them in terms of the nitrogen bases, the A, T, G, and C of DNA, or the A, U, G, and C of RNA. Each nucleotide has three parts. One is a phosphate group. I mentioned that phosphate sugar backbone. When we're talking about DNA, the sugar is deoxyribose. When we're talking about RNA, it's ribose. And then probably the part you're most familiar with are the nitrogen-containing bases. The letters that stand for them are A, T, G, and C. Adenine, thymine, cytosine, and guanine. And we'll get to those names in just a second. You don't have to worry about writing them down. So here we have we have a nucleic acid, which is a nucleotide chain. Each nucleotide is a phosphate, a sugar, and a nitrogen-containing base. And when we add them in a dehydration synthesis to one another, we get a nucleic acid. So the nitrogen-containing bases, these are what in DNA we call the genes. This is where we get the information of what's a capital T, what's a little t. The things that we talked about in terms of genetics in the last unit. So, for example, height. When we talked about height, and we talked about the dominant trait of tall. Tall was represented by two alleles, capital T and capital T, or capital T and little t. And the recessive trait of short was represented by two little t's. Remembering again, you have two alleles for every trait because you get one from biological mom and one from biological dad. So let's say that every three nucleotide bases, an A followed by a T, followed by a G, it represents at that gene locus, the gene location. When you see an ATG, it represents a capital T. When you have GCC at that gene location, it represents a little t. So what people do is they study strands of DNA and they see repeating things, and then they're like, oh, and they figure out this puzzle of when you have these 3 in a row, it means this, and when you have those 3 in a row, it means that. So let's say that you have for someone who's homozygous dominant, capital T, their nucleotide strand at that gene locus is ATG. So we know that ATG codes for capital T, and when you have this strand repeated, it stands for being homozygous dominant. If you are heterozygous, capital T, little t, your nucleotide strand at that gene locus would be an ATG for the capital T, followed by a GCC for the little t. And if you are homozygous recessive and show the recessive trait, you are little t, and each little t is represented by a GCC followed by another GCC. So taking what we talked about in the last unit and synthesizing it down to more of the, what are the actual nucleotides saying? This is how we connect that information together. So now, DNA is two-sided. We're just looking at one side here. When we have two sides of DNA, we have one that are the coding portion, the coding side of the DNA, and the other one is a complementary side. So let's talk about the base pairings. The 4 bases, as I mentioned before, A for adenine, G for guanine, C for cysteine, and T for thymine. They pair together. Of the 4 of them, 2 pair together and the other 2 pair together. So it's called base pairing because adenine always pairs with thymine, and guanine always pairs with cysteine. So again, A always pairs with T. They are base pairs. And G always pairs with C. Even if there's a mutation and an A is replaced with a G, the G will then base pair with C. So what are these? They are called purines and pyrimidines. The bases are, one is a little bit bigger than the other one in the base pair. So you have one that's a little bigger, the little bigger one is the purine, and the smaller one is called a pyrimidine. A and G are the little bit bigger ones called the purines, and T and C are the smaller ones, the pyrimidines. The way the base pairs come together is with hydrogen bonds. They are hydrogen bonded together. Remember that when we were talking about hydrogen bonds, that a single hydrogen bond is not very strong, but when they are in chains, like you have a whole long chain of DNA, it makes this very strong. It gives strength to the DNA. So regardless of how many A's you have, you will always have the same amount of T's because they go together. And how many G's you have, you'll always have the same amount of C's because they are partners or pairs. And this is just to give you an idea when I say one is bigger and one is smaller, and the purines are bigger and the pyrimidines are smaller. What I mean by that is, like when we look at, for example, adenine and thymine here, you can see that on all of them, the phosphorus is the same, the sugar, deoxyribose is the same, but the bases are different. And when you're taking a look at the purines, the adenine and guanine, they're just a little bit bigger in that they have 2 rings to make them up, whereas the smaller pyrimidines, the cytosine and the thymine only have 1 ring. So they're not like grossly bigger or smaller, just a little bit. All right, the order of nucleotides in the DNA can encode for so much information. We can pack so much information. We've been saying that all along into our cells. It's not necessarily the amount of DNA bases, of nucleotide bases that you have that's important, but it's the sequence that they come in. You remember back in the last unit, we talked about the number of chromosomes that humans have. Our diploid or 2N number is 46. And I mentioned crayfish, which is like a little tiny freshwater lobster, that they have 200 chromosomes. They don't even have a full brain like we do. They're a lot more simplistic. So they have a lot more DNA than we do, but does that mean that they're any more complex? No, not necessarily. Ours is just a little bit more efficient. We have less DNA, but the sequence that everything comes in is very efficient, and that's what's more important. When you're looking at a bacteria, even a single-celled organism that doesn't even have a nucleus, they have millions of DNA bases. And when we're talking about organisms like us, or plants, or fungi, we're talking about billions, or even probably more like trillions of bases. When we take a look at the structure of the DNA is, if we unwind the DNA, going from a helix and making it flat, it is like a ladder, where it's got two sides and then it's got rungs in the middle. The nitrogen bases in the middle are like what you would climb up the ladder. The sides that you would maybe like, the sides that support the ladder, that's the phosphate sugar bases. So here, when we take a look at our DNA helix, and we don't have much detail, when we take a look at the rungs of the ladder, the rungs are the base pairs. And when we take a look at this in a little bit more detail, the bluish purple part here is the phosphate sugar backbone. So that is a repeat of phosphate and sugar all the way there. And in the middle, you have your base pairs that are connected by hydrogen bonds. I'm going to get to that later. Okay, so let's talk about replication of DNA. When you replicate the DNA, like we talked about prior to going into any kind of cell division, the DNA will replicate. We're going to talk about just a little bit of details on how that actually happens. So we know that DNA replication happens in the S phase of interphase of the cell cycle. It happens prior to, if we're talking about mitosis or maybe meiosis, or binary fission if we're talking about a prokaryotic cell. The DNA replication happens in the nucleus, and then going into the prophase, the first phase of mitosis, or prophase 1 of meiosis, what we know happens to the nucleus is the nuclear envelope starts to break down. The DNA goes from all unwound and loose chromatin, and it wraps around histone proteins to become linear pieces of chromosomes. When the DNA replicates, each chromosome, upon replication, gets a genetically identical sister chromatid.
It's not.
Did I skip that one sentence?
Yeah, I think. No, it's right here, right? This is.
Oh, DNA replication is called semi-conservative replication.
Oh,
There you go. I think the nucleus one was filled in for you.
yeah.
Yeah, sorry about that. So when I talk about semi-conservative replication, we are conserving the DNA message in that when you have, is it messed up? Did it? Oh, yeah, okay. I just highlighted something that shouldn't have been highlighted, I think. Sorry for the confusion. So if these are the original homologous pair of chromosomes, right? Let's say yellow comes from biological mom and purple comes from biological dad, that when the cell is starting to enter into mitosis, meiosis, or binary fission, that each of these original homologous chromosomes gets a genetically identical sister chromatid that is temporarily glued together by these what we call centromeres. The original message is in this side of the chromatids, and the new copies are on the outside. So what we mean by semi-conservative replication is that we are conserving the original message, but we're adding a new copy to go into other cells. So to start, what happens is that we have our DNA, and it's all wrapped around itself. The DNA will start to unwrap, and the nucleotides here, they have hydrogen bonds between them. You will have an enzyme called DNA helicase that will attach to each chromosome, and it will start to go chop, 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 and it'll chop apart the double-stranded into single strands. So that's what DNA helicase does. Following DNA helicase chopping apart the double strands into single strands, is on each of the strands that's opening up, you have an enzyme called DNA polymerase. So how the nucleotides are like this, and then DNA helicase pulls apart them at their hydrogen bonds, it breaks that hydrogen bond. A DNA polymerase goes onto this side of the strand, and a DNA polymerase goes onto that side of the strand. And what the DNA polymerase does is if this is A and T, the DNA polymerase on each side, when these are pulled apart, the polymerase reads A, and then floating out in the cell are individual nucleotides of A's, T's, G's, and C's. And when the polymerase reads an A, it pulls a T and it adds it over here. And then on this side, the one that opened up, it had a T, and the DNA polymerase on this side is like, oh, I need an A to go with this T. Polymerase is just reading each of the nucleotides that's pulled apart to add a new copy who matches the base pairing of A to T and T to A. Then you have DNA ligase, and it's going to glue together the new nucleotide pairings. So you have three enzymes here in terms of replicating the DNA. One is DNA helicase pulls apart the base pairs. It breaks apart those hydrogen bonds. Chop, 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 chop. DNA polymerase reads each of the nucleotides that is then separated, and it calls over the appropriate base pair. And DNA ligase will put that hydrogen bond back to make that new base pairing stick. Each strand now contains one of the original parts of the DNA, and it's added a new part on the inside. So when we're using my example like this, and DNA helicase comes and separates them, this part here, these are the original parts of the original chromosomes. And DNA polymerase here and DNA polymerase here are making and adding a new strand on the inside. So each of your new strands, you have the original old strand, and DNA polymerase adds on a new strand, and over here you have the old strand, and DNA polymerase adds on the new strand. So you end up with two new chromosomes, an old strand, and a new strand. So for example, if we are taking a look at just something simplistic like this, what happens here with our parent DNA is that DNA helicase comes in and chops these apart. Chop, 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 chop. Then what happens is helicase will bind onto this first G. No, sorry, excuse me. Polymerase. Helicase chops the hydrogen bonds. A polymerase will add on here, and a polymerase will add on here. This first base pair of G and C, you can see the outer part is this strand, the inner, I guess they're both outer to you, but this strand here begins over here with C. Polymerase will read these, each gets a polymerase. Polymerase will read the G, and it'll call over a C nucleotide, and then ligase will make this new hydrogen bond. So this side, your left, maybe I should say left side strand is here, with a new right strand added on by polymerase, and your right strand over here of your original is here, which has a new strand just like the original left strand here. This is the way it actually happens. This is a bit more descriptive. So DNA helicase comes and it chops apart. It'll start, way down here at the beginning. When we get to this part, DNA helicase is coming and it's chopping apart. It will continue to chop these apart. To break our DNA double strand, it's going to start making it into 2 strands. DNA polymerase is going to read all of these open strands. So if we're at this point, DNA polymerase is going to be attached here, and it's going to call over an appropriate base pair T2A. And then it'll read A and call over a T, and G and call over a C, and G and call over a C. It's going to be doing another polymerase. It's going to be doing the same thing on this other side of the strand. All right, so remember, we said sometimes mistakes do happen, that the volume of DNA being copied, and all of these actions happen so quickly that sometimes mistakes do happen. When we're talking about a mistake in the copying of the DNA, it's called a mutation. Mutations are more significant when they happen in the sperm and egg, because then those mutations are given to the offspring. So meiosis, remember when we're talking meiosis is the cell replication of making sperm and egg. And that's where mutations are most significant. Those mutations are then passed on to the offspring. So different kinds of mutations can happen. We're going to look at just some really simple examples. One could be a point mutation. And in a point mutation, one nucleotide is changed in the entire strand. So if we take a look at our original DNA strand here, we had AACG originally, and there's a mistake, and instead of putting a G there, they put a C there. That's a point mutation. Next kind of mutation is called an insertion mutation. One or more nucleotides are accidentally inserted into the chain. So if we're looking at our same strand, we had AA, and where that C should be, accidentally inserted is an extra 3 nucleotides. You would see that the DNA strand goes from this, and it is a little bit longer because it has 3 extra nucleotides added. And then a deletion mutation is when one or more nucleotides are deleted from the strand. So in our strand after that AAGC, we had a CA, and they are missing from the final outcome. We'll come back to this in either a little bit or next lecture and talk about what are the outcomes when these happen. All right, so how are genes interpreted? Remember that there's 2 alleles for every trait. There's 2 alleles at a gene. When we're talking about what 2 alleles those nucleotides code for, those give us 2 genes or alleles, 2 alleles for every trait, right? And when you have 2 alleles for that trait, that codes for your genotype for that gene. Then we take that information of the genotype and we translate it into what does it mean physically? Does it mean tall or does it mean short? And so then the outcome of what your genotype is, we describe, well, what does that really mean to the organism? How do they look? How does their internal physiology work? What behaviors do they do? Those are the phenotypes. So we know we've been concentrating on the outcomes of our genetic problems for looking at the genotypes of the offspring, the possible genotypes and possible phenotypes of the offspring. All right, where does protein synthesis occur? Do you remember where? Did protein synthesis occur in the nucleus? Was it on the smooth endoplasmic reticulum outside the nucleus? Was it on the ribosomes outside the nucleus? Or in the Golgi body outside the nucleus? Does anybody remember what organelle protein synthesis occurred at? Yeah, good. Okay, so the protein synthesis occurs on the ribosomes. This happens outside of the nucleus. Now, the DNA is inside the nucleus, and then protein synthesis happens outside the nucleus. So we're going to have a process of getting our DNA from the nucleus to the ribosomes. What we have to get there is we have RNA. DNA has the instructions for a gene. RNA takes a copy of that instructions in its own kind, which is RNA. It's a little bit different. And then it goes to the ribosome, and with the help of the ribosome, the message from the DNA that is in the RNA creates proteins. And then proteins can be changed into other proteins. It can be changed into lipids. It can be changed into whatever the cell needs, whatever is coded for in that DNA. So RNA is similar to DNA, but it has a few differences. One is RNA is single-stranded. Remember, DNA was double-stranded. RNA has the sugar ribose instead of deoxyribose. That's nice, it's right there in the name, right? The sugar for DNA is deoxyribose, the sugar for RNA is ribose. And here's one more thing about the nitrogen-containing bases. We don't use a T in RNA, we use uracil. So A pairs with U pairs with A. G and C still go together. G and C do not change in RNA, just RNA uses uracil now. There are 3 different kinds of RNA. It's not just one kind of RNA. We have one kind of DNA, but we have 3 different kinds of RNA that help us get to that final protein outcome. So here's all the 3 different RNAs. One is going to have the original message of the DNA. This is called mRNA. The second kind of RNA is part of the ribosome. We call it rRNA for ribosomal RNA. And then tRNA helps to bring the amino acid parts of the protein that gets made. The whole thing is that these 3 different RNAs are very important in making the amino acid sequence that makes up the final protein product of what was in our DNA instructions. Because each base pairs with a complementary base in every DNA molecule, the amount of, would cysteine equal guanine? Would cysteine equal thymine? Okay, good. I see some no's at least on that one. Would cysteine equal adenine? No, they don't go together, right? Each nucleotide is unrelated to all the others? No, we know when they base pair. So, what's the correct answer? Okay, number 1. Okay. Okay, so cysteine, regardless of how many nucleotides you have, you're always going to have an equal amount of cysteine and guanine. You're always going to have an equal amount of adenine and thymine. Oh, you don't have this. I always forget to put this in there, but so just to note, this whole thing is called the central dogma. Chapters, the rest of this chapter, but really chapter 15 and 16, in biology we call this the central dogma. It's taking the information in DNA it's making a transcript or a copy of that into RNA, and it's making the final protein product. Yeah, if you want to write it down or you want to take a picture, I'm not going to put this term on the exam, but at least when you hear it in the future, the central dogma, it just means this process of going from DNA to RNA to the protein product, just so you know. This overall idea of the central dogma, it takes the information, the instructions in DNA, and it helps us get to whatever the cell needs at that time. So like if you're an eye cell and you need to replace an enzyme for seeing in your eye, you're going to read that gene in the eye cell. You're going to take the information, the instructions in the DNA for that eventual enzyme. It's going to be copied into an RNA transcript, and then you'll make the protein. That's the whole idea. Now, the instructions, they don't just stop there, that if you need this enzyme and it needs to be reconfigured or changed a little bit from its original DNA instruction product, that protein that is made might go to the endoplasmic reticulum to finish up what needs to be done to it. Maybe add some glycoprotein attachments to it. So how does this information happen? The central dogma is this. You have the DNA undergoes transcription. We're taking the information in the DNA and we're making an RNA copy. We call that a transcript. Just like, let's say in this class that when you have been taking your exams all semester, I'm taking what you earn on the exam and I type it into the computer, right? I'm not taking what happened on your actual paper, I'm making a copy of that grade into the computer, and then at the end of the semester, we're gonna add up all your grades in the computer, and I'm going to enter your grade into a system. And then that grade entered, all of your grades that are entered into this system for the college, they go onto your transcript. And that's a copy of how you've done over time at your college career. So I'm taking the work that you've done and you're getting a transcript, you are getting a transcript from the college. So you're making a copy of your work into one place. It's kind of like DNA is making a copy into RNA. RNA, that mRNA, we'll talk about that in just a second, which stands for messenger RNA. It leaves the nucleus and it goes out to a ribosome where the transfer RNA is going to bring over the appropriate amino acids. And at this translation point, all these amino acids add up in a chain to create a protein, which is the final product of this central dogma. Now, we're going to walk through this process. We'll do a little practical example of how this actually happens. Remember that DNA has 4 nucleotides. A pairs with T, G pairs with C. However, when we are taking the information from DNA and we're putting it into RNA, A will now pair with U. G and C still pair together. So just keep that in the back of your mind and I will remind you too. Okay, so let's do an example of this central dogma here. Let's say that this is the original DNA strand. So what I want you to do is write out the template strand. DNA is double-sided, and when we're talking about, well, if we have two sides, which one becomes the instructions for the RNA? The one that becomes the instructions of the two sides is called the template strand. So I want you to write out A, what is its complement? We're talking DNA to DNA. So I'll get you started. A complements U. No. A complements T. A and T go together in DNA. T complements A. G complements C. C complements G. So continue on, remembering which nucleotide complements what the other one is, its appropriate base pair, and make that template strand. So again, remember, A complements T, T complements A, G complements C, C complements G. Your DNA template strand, hopefully, you got this. And you'll also see it is listed below. So what happens is one of the sides of the DNA is marked within the DNA that this is the template strand. So here's your template strand again. Now, what you are going to do with this is you are going to make a complementary mRNA. Remember, when you're going from DNA to RNA, think what complements what? T complements A, T pairs with A pairs with U, C pairs with G, G pairs with C. So T complements A complements U, C complements G, G complements C. So continue on. Just, you have to remember that A will complement U. T still complements A. Your mRNA strand will look like that. This one's just, you have to keep it in your brain that T complements A complements U, C complements G, G complements C, C complements G, G complements C. A here, again, complements U, C complements G, G complements C. You've got these two T's, they complement A's. These two A's complement U's. And then you have a T complementing an A and a C complementing a G. So things just get a little bit different. And just really think in terms of what complements what or what pairs with what. Now, we're taking our mRNA strand. Remember that this has the message of the DNA. This is the transcript. It has that original information that DNA had. It's just a little bit different. It's single-sided. It has the sugar ribose, and A complements U now. So we're going to take our mRNA strand, and you're going to make your complementary tRNA. It's RNA to RNA. A complements, so we start out with this one. A complements U, U complements A, G complements C, C complements G. There's no more T's, no T's at all, because we're going RNA to RNA. So remember, A complements U and U complements A. C and G still go together. So in taking a look at our tRNA strand, A complements U, U complements A, G complements C, C complements G, G complements C, C complements G, U complements A, G complements C, C complements G, A complements U, A complements U, U complements A, U complements A complements U, and G complements C. Okay, at this point, what you would do is you would, to get the amino acid strand, is every 3 nucleotides, they work in what are called codons. Every 3 nucleotides code for an amino acid, but we look up the mRNA. So this is a big thing to remember, and you might want to just, and we're going to get into this in a little bit more detail shortly, but you might want to circle the mRNA and say, this codes for the amino acids. The mRNA codes for the amino acids. The mRNA codes for the amino acids. Don't forget, mRNA codes for the amino acids. The mRNA. You're always going to look up, when you have the amino acid chart, you're going to look up the mRNA codons. Every 3 mRNAs code for the nucleotide, code for the amino acids. So, for example, and you'll see that chart when we get into lab, that when I go to that chart and I look up the mRNA 3 nucleotide codons, and I start with AUG at the beginning of my mRNA transcript, when I look that up, I find methionine, and it is what we call a start codon. At the beginning of every amino acid, we should see an AUG sequence. Our next 3 mRNA codons are CGC. I look them up and I get arginine. These are all different kinds of amino acids. When I look up the next 3 mRNA codons, it's UGC, codes for cysteine. When I look up AAU, our next 3 mRNAs, I get asparagine. And when I get to the end, UAG codes for a stop codon. When you look at an entire DNA gene and it's translated into mRNA, the mRNA copy of the DNA message will start with AUG, and then there's 3 different stop codons. And so one stop codon, for example, is UAG. So you always see a start codon at the beginning and a stop codon at the end.
Everything be in the middle, be like swap.
Yeah, oh yeah, yeah. You've got the other, there's 20 amino acids, so yeah, you'll have a variety of, depending on the sequence of these, will tell you what the outcome protein is. Yeah, good question. I don't know. Okay, so I think
Chapter 15 and 16.
okay, so now we're going, yeah, good. Okay, so now, let's take a look. All right, you have this question?
Yeah.
Okay, good, okay. So when comparing DNA and RNA, we find that no sugar is present in either molecule? Yes or no? No, they have deoxyribose for DNA or ribose for RNA. Only DNA has a backbone of sugars and phosphates. Well, we don't even know because I haven't talked about that, but let's skip that one. Adenine pairs with different bases in DNA and RNA. Right, adenine pairs with thymine in DNA and adenine pairs with uracil in RNA. Thymine pairs with different bases in DNA and RNA. Thymine always pairs with adenine. So number 3 is correct. All right, so let's talk about this whole like making genes and the expression of genes. So how does the information in DNA translate to RNA? This again, we call it the central dogma. You have these required steps of the DNA undergoes transcription. The message of the DNA gets a transcript or copied into mRNA. Our next step is translation. The tRNA pairs up with the mRNA to undergo translation of that message, and it makes are final product of an amino acid sequence that makes up a protein. So this is, we're going to go through the process in a little bit more detail. This is essentially the last part of what you need to know for the exam. So the DNA is in the nucleus. Transcription happens in the nucleus. The mRNA transcript goes out of the nucleus. It meets tRNA and the amino acids here on a ribosome. All right, so sequence of bases. As I mentioned before, something about this codon term. A codon is 3 nucleotide mRNA, and they code for a specific kind of amino acid. So again, mRNA codes for the amino acid. mRNA codes for the amino acid. You definitely want to remember that so that you don't get that confused with the tRNA. There are 64 different possible combinations between the A, U, G, and C's. So there are 64 different codons, but this is a really cool thing that helps our cells to make less mutations. There's 64 codons, but they only code for 20 amino acids. So you might be like, okay, I'll tell you more. Each codon, each of these 64 codons equates to 20 amino acids, which means that some different codons code for the same amino acid. So you might have 4 codons that are different, but they all code for the same amino acid. And then we'll look closely at, like, still, how does that eliminate or lessen the amount of mutations? The codons here also account for 1 start codon, as I mentioned, and 3 stop codons. You'll always see that AUG at the beginning of a protein sequence, and you'll see one of the 3 stop codons at the end. Let me show you. I don't know why I don't have this in there, but I should. Okay, so you have this in your lab book and you'll be working with it today, that you've got this like chart. These are all the 64 codons. So let's look, for example, let's look at proline. That when you have CCU, CCC, CCA, or CCG codons, all of these combinations code for the same amino acid. So if there's a mutation and you go from a CCU and the mutation changes that last amino acid in this codon to a C instead of a U, that mutation still codes for the same thing. So this is what we call redundancy, that you have often the same 2 amino acids in a codon, and the last one is different. So that if you have a mutation to the last nucleotide in the 3 codon sequence, you'll still get the same outcome. So a mutation can be a nothing. Like, yeah, you have a mutation, but it does nothing for that cell. It means the same exact thing. So that's a good thing, that we have this redundancy. You can see again, like these ones here. that you have CCU, CCC, CCA, and CCG. They all code for alanine. Yeah.
On the exam, you have to know like each one.
No, I would give you this. Yeah. No. Do not memorize this. I would, if like in that last example where I gave you, had you work from the DNA template to the mRNA message and then said, based on this mRNA message, what is the 4 codons, what do they code for? I would give you this chart. And so you wouldn't have to memorize it or anything, you'd be able to look it up on the chart. Yeah, good question. Yeah, because that looks a little like overwhelming.
Are these kind of mutations really common?
Yes, they are common. Not like super duper duper common, but more common than some of the more, the larger mutations, where you have like a huge insertion or a huge deletion. It's often like one amino acid, and then that happens and you're like, okay, whew, and it doesn't do anything. Yeah. Simple. These are simple mutations which are more common than the more complex ones. Yeah, good question too. So just to show you like what I'm talking about and all this, we'll work with this codon table in lab. But just remember, you are always looking up the mRNA, always the mRNA, not the tRNA, the mRNA. Okay, so let's talk about transcription. So we're taking that information in transcription, we're taking the information in the DNA, and we're making an mRNA transcript, or a copy of that information. So DNA is transcribed into mRNA. Remember, this happens in the nucleus. And when the DNA sequence is read, and you are making the complementary mRNA, you have what's called RNA polymerase. If you remember when we made a copy of DNA to DNA, when we did DNA replication, we used DNA polymerase to add on the free nucleotides that complement each DNA nucleotide. So now when we're going from DNA to RNA, we use RNA polymerase. Polymerase is an enzyme that is going to grab free nucleotides and add its complement on either DNA to DNA for DNA polymerase, or DNA to RNA, or RNA to RNA, we use RNA polymerase. Okay, so what happens is the area of the DNA that we need, the gene, whatever that product is that we need right now in our cell, the DNA is going to untwist, and it's going to separate the template strand. There's like a marker in the DNA that says this is the side that we want to make the mRNA transcript of. And the RNA polymerase is going to attach to an area that's called the promoter on the DNA. And that's going to make the DNA go, ooh, okay, I'm going to unwind, and I'm going to start to unzip my hydrogen bonds between the two nucleotides. And the RNA polymerase, based on information in that promoter, that area of the DNA, it knows I'm going to go on this side because this is the template strand. So RNA polymerase is going to work its way down the DNA, and it's going to add on complementary mRNA bases to the DNA template strand until it gets to a terminator sequence. And the terminator sequence holds the stop codon, and it says, okay, you're done. RNA polymerase, you're all done. You don't need to add any more nucleotides. We've got that information, that transcript of the DNA instructions in RNA. The terminator sequence says to the RNA polymerase, go away. It releases the mRNA transcript strand, which is going to be stimulated to swim out of the nucleus and go find a ribosome. And then it causes the DNA to go back to being zipped and wound. So it's kind of like this, that if this is the gene that we need, the RNA polymerase is going to attach and it's going to read this side of the DNA. But in the DNA here, in this area, there's a promoter sequence that says, hey, RNA polymerase attach here, specifically read this side. This is the template side. That's what the promoter area over here of the DNA tells the RNA polymerase. The RNA polymerase reads each of the DNA nucleotides, calls over the appropriate mRNA complement nucleotide, and it reads until it gets to the terminator sequence where you have a stop codon, and it's like you're done. That then stimulates the DNA to go back to being hydrogen bonded back together, and it winds back up. The mRNA is going to leave the nucleus, go find a ribosome, and then begin the translation process. So mRNA is also processed. We have a lot of, in our DNA, we have a lot of what's called junk DNA within our chromosomes. We translate this information into exons and introns. So within that mRNA transcript, not all of it is useful to make the protein. Introns are removed. They get chopped up. They get spliced out. The exons leave. So if you're like exon, intron, the introns are what we would call a junk portion of the DNA. The exons are the important stuff. You can remember that perhaps by remembering that exons exit the nucleus. If they are the mRNA portion that is going to exit the nucleus, that is going to go to the ribosome and be read into protein. So the exons that exit, those are the coding portion. The introns are the junk portion. To get the introns out, that junk portion, you have an enzyme that's called a spliceosome that is used. Again, the introns are the junk DNA. All right, this is the weird thing about our DNA as opposed to maybe like that crawfish that has 200 chromosomes. Within any area of a gene, we have to pull out some of these introns before it gets processed into a protein. The areas that get pulled out, the introns, they may be junk for, let's say, an eye cell. That an eye cell, we need to make a protein that helps us to see the red color. But within the original DNA template strand, there's information for making a heart protein. So in the eye, if we want to make eye proteins that help us see red color, we don't need the heart parts of the DNA instructions. So we cut out the heart parts. Now, junk DNA does not mean that it's junk to every single cell. So that heart information is junk to the eye cell, and to the heart cell, the eye information is junk. We have not as much DNA as other organisms because we have these weird, like junk portions interspliced with coding portions in our cells. And so this is a lot of the puzzle of figuring out what do these areas of DNA mean and what do we need to pull out for certain cells as opposed to other cells. Yeah, it's like very complicated. All right, so remember the exons exit the nucleus and code for the protein because they go and exit the nucleus to go to the ribosome where the protein is made. All right, these spliceosomes, they're an enzyme that cuts or splices out the introns. good name for a enzyme that cuts something up, a splicer, a spliceosome. So if we take a look at, if this is our original DNA area, and the lighter pink colors are the ones that actually code for the protein that that cell in particular needs, but the darker portions the cell does not need, you would have spliceosomes that splice out these introns. So when we're taking a look, that we have an exon, then an intron, and an exon, and an intron, and an exon. And these would all be our exons. And the spliceosomes would splice out these junk intron areas for that cell.
Is there a reason why the exon is bigger than the intron? Or is it just like that?
At the end,
No, like over there too. In the initial RNA transcription.
here,
Yeah.
why there's more exons than introns. Just for this cell, yeah, sometimes you might have more introns than exons. It just depends on that particular cell and that gene. Yeah, good question. Okay. All right, let's take a look at this question. Which of the following could explain why a mRNA molecule would become shorter before it is used in translation? The cell uses enzymes to cut amino acids out of the mRNA molecule. Is it cutting amino acids? Not amino acids, that's what it's gonna make later. The mRNA molecule needs to be cut into pieces in order to fit into the ribosome. I didn't say anything about that. Enzymes in the nucleus remove introns from the mRNA molecule. Yeah, they're removing the introns. Enzymes in the nucleus remove the exons. No, we want to keep the exons. We remove the introns. So number 3 is correct. All right, translation. Our protein synthesis. mRNA is going to get together with tRNA. mRNA still has the original message of the DNA. Right? We said that mRNA codes for the amino acid. The tRNA is the translator. So the mRNA has the original DNA message, the codons. The tRNA is only the translator. tRNA has the little amino acids on their back. So every 3 tRNA nucleotides are called anticodons. And they have a little amino acid backpack. It's like as though the nucleotides of the tRNA are sushi rice and the piece of fish is the amino acid, that they're glued on together. And the sushi rice is going to bring over, the tRNA is going to bring over the amino acid, the fish, to the mRNA. But the mRNA says, this is who I need. The tRNA is just like, okay, here, let me bring that to you. So, role of tRNA. They're attached to the amino acids. Again, like the tRNA is like sushi rice, and the amino acid is the fish. They are glued together. They come as a pair, but they are bringing what mRNA is asking for. We call the mRNA the codons because they are coding for the amino acid. The tRNA are called anticodons because they are not coding for the amino acid. I like when they have nice names in biology, that makes sense. Okay. There's at least 20 different tRNAs to carry the 20 different amino acids. But again, we have different codons that code for the same thing. So we know that an amino acid can be coded for by more than one codon. We saw that redundancy in the mRNA table.